related to mitosis and meiosis. Okay. A little bit of follow-up information from last class, things that are important but we didn't quite get to. And there's going to be a little bit of a change in terms of structure of the class, this class, that I hope will be useful. So this is the famous DNA replication DNA sequence that has some confusing stuff in there. How many TAs are there and so forth. You furiously tried to copy this down a few classes ago. And I asked on the slides for last class to rewrite this using the microsatellite notation. So one of the topics or one of the tasks that I had for the last class about mutation was using a specific type of notation to describe microsatellites. Okay, so what's a microsatellite? It's not a tiny little thing that spins around the Earth and gives us cellular service. What's a microsatellite when we talk about genetics? Videos from not the ones you were supposed to watch for today, but the ones from the previous class.
It's an indel using repetitive pieces of code.
It's basically, essentially, it is a type of DNA sequence that's one of these stuttering events. Like when there's a dinucleotide, two nucleotides, that was one of the vocabulary items. Dinucleotide, trinucleotide, tetranucleotide. That series of 2 or 3 or 4 nucleotides in a row that are back to back, just repeated over and over again. So how many TAs are there in this microsatellite? So a microsatellite is just a repeat, one small DNA sequence that's repeated over and over again, head to tail. 7. So the way you would write this in shorthand, if you were trying to copy this down quickly and you were trying to do this a little bit faster, is how would you write that in microsatellite notation? Starts with the single unit. Then, yes, it's in parentheses. And then there's a subscript with the number of repeats. So that tells us this is the unit that's repeated 7 times. It's TA, 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 TA. Just that easy. Since we're talking about formatting stuff, does anybody remember how to write a FASTA DNA sequence? If we were going to turn this into a FASTA formatted sequence, what would we have to do?
Put a greater than sign first.
We'd have to put that sign and then some sort of descriptive name, which is totally up to you. So if I gave you just a sequence of DNA and I asked you to turn it into a FASTA formatted sequence, that's what you would need to do. The greater than sign, and then give it some descriptive name. That would be up to you, what you called it. In this case, it's a random sequence I made up, so it has no name. Are there any other microsatellites or ways that you could use this shorthand in this sequence to shorten the writing of this entire string of nucleotides? There's what? Okay, so we've got GAC-TAC, which I don't think I realized I was doing when I wrote this, but apparently I thought I was just randomly hitting G's and A's and T's and C's. That's what I do. I just hunt and pack and just... If I accidentally hit one of the neighboring keys on the keyboard, I just delete it. So I get some E's in there too. I just delete the E's. But maybe I accidentally copied and pasted or something. Because when I looked at this later, I realized that there were a lot of those GAC, TACs in a row. So how many there? Also 7. If I count correctly. So you could write in, after those TAs, there'd be an AGTC, and And then what would we write?
Cac-tac, 7.
Yep, cac-tac 7, and so on. Now, I would not agree to doing this, only because this is not a microsatellite. A single nucleotide being repeated,
It'd have to be 2 or more. Right?
like here in yellow that I'm highlighting. Those T's, you couldn't say that's T parentheses 8. That's not a single T is not a microsatellite. It needs to be 2 to 3, 4. There's probably an upper limit that I don't know what the definition of the upper limit of a microsatellite is, but that's the idea. Yes?
So to simplify that, we should just do TT4?
Yeah, if you want to. That's totally demonstrates to me that you understand the concept. You could say that instead of me complaining that that's not a dinucleotide repeat, it's a perfect argument. You could say, well, I think that's a TT repeat. So instead of T8, you'd write TT4. Now it's a microsatellite. That's totally fine. Okay, so that's microsatellite notation. Good, everybody's almost done with that quiz. Now, a little bit more from last class. I just want to talk really briefly about mutations. You don't need to memorize this information. Don't focus on the chemical reactions that are happening. Just focus on the big picture. I have to worry about going out in Fresno generally because I have no pigment. As you can tell, I'm Scandinavian, I'm British Isles and Norwegian, Swedish. If UV hits me, it damages the DNA in my skin cells. Maybe it penetrates a little bit below the skin cells, but that's about it. What happens when ultraviolet light hits DNA? Mutation, but it's not the ultraviolet light hitting the DNA that damages the DNA that causes the mutation. That's the weird part. So what's showing up here is what happens when high-powered photons hit DNA. If there are 2 thymines that are right next to each other on the DNA, a TT, like in that microsatellite we were just talking about, or not microsatellite, depending on your point of view, ultraviolet light can actually cause those 2 thymines to covalently bond to each other. And that makes a little kink, sort of, in that backbone of the DNA strand. The problem with this is mutagenic not because of that covalent bond. That's just one strand of DNA. That's not important. What's important is that our cells, which have evolved over time to deal with things like ultraviolet light hitting our DNA and destroying our DNA, our cells encode these enzymes, like polymerase 4 and polymerase 5, that are specifically designed. Those proteins go around our DNA, binding the DNA and looking for, feeling for, sorry, I'm anthropomorphizing the proteins, they don't have eyes, feeling for those diagnostic sort of kinks in the DNA backbone that suggest that, okay, now I have felt on the DNA that there's a thymine dimer, which is what these are called, when the 2 thymines link to each other. What does that enzyme do when it finds it? It doesn't repair the problem. What does it do? What does this cartoon show it doing? When DNA replication happens, there are 2 T's there. They're dimerized. What's being synthesized on the new strand by these fancy DNA polymerases we have that are meant to solve the problem? GA. Does that make sense? What happened? What just happened when that DNA polymerase that's supposed to be solving the problem of thymine dimers copied that strand?
Caused an SNP, so in the new strand of DNA, there will no longer be 2 T's next to each other, and it won't keep in the future, or it won't bind in the future.
Right. So the good thing for our cells is that at least a DNA polymerase has been able to copy the chromosome. It's been able to replicate the second strand, so we've got a new double-stranded chromosome. The problem is that now we've got a template that has G and T in it, or G and A in it rather, which means the next time DNA replication occurs, when this strand is used as the template, you've got a G and an A there, what's the new strand sequence going to be? There's going to be a C there and there's going to be a T there. That's where the mutation happened. Did you see it? The original DNA strand had what on the top strand? 2 T's. They're the ones that got damaged with the ultraviolet light. Now what do we have? CT. Because this DNA polymerase doesn't have any template to read when those thymines are dimerized. This polymerase can't tell that there are 2 T's there. So it doesn't know to stick in 2 A's here. It sticks in random nucleotides. This polymerase, this protein that's in our cells that is meant to be a polymerase, an enzyme that replicates our DNA for us, is error prone. It's designed to just put in whatever nucleotide it can because that's better for the cell than the cell not being able to replicate the chromosome at all. If a normal DNA polymerase was trying to replicate through that thymine dimer, it would get stuck there. Synthesis of the second strand would stop, so cell division stops. In cell biology, if you don't divide, you're dead, basically. The whole point of being a cell, most of the time, especially during development, is to grow and divide. So overall, better for the cell to mutate the DNA and to be able to finish cell replication, cell division, DNA replication, than it is to have a mutation. So that's why ultraviolet light is mutagenic. Damages the DNA. It's the repairing of the DNA after that damage that causes the mutations.
So is there a chance that if they're just independent base pairs that they could end up getting it right?
Yeah, absolutely. And we could let's calculate the probability of that happening. What's the probability of sticking 2 adenines next to each other if you're just randomly inserting nucleotides on a DNA strand? You got it right.
Does the kink have any effect? Like if they are, the correct random nucleotides are placed, but they're still.
So that's only one, yes, there is still a kink in that one strand of DNA. The good news is that the strand that's newly synthesized doesn't. So after the first cell division, you have one kinked strand and one normal strand, but then they become templates for making one. Now you've got one normal double-stranded here on the bottom and a kinked plus normal strand on the top. And every additional cell division, you make more regular non-kinked templates. Because every additional cell division, those strands separate and become the templates for the next round of division, the next round of replication. So eventually you basically dilute out the effect of having that thymine dimer, but it never gets repaired, to my knowledge.
Another question, when the UV light hits, is it only affecting 2 nucleotides or does it affect much more?
It just affects those 2 nucleotides. When a photon of high-powered light hits, it causes the formation of that bond in that one nucleotide pair.
No such thing as adenine dimers in UV light?
Something about the organic structure of thymine that I don't remember offhand that makes them particularly susceptible to doing this. So right, to get back to this quickly, to this earlier question, yeah, there's a 1 in 4 chance that you would, by chance, that polymerase would stick an A there. And then there's also a 1 in 4 chance that it would happen to get another A next to it. So you've got a 1 in 16 chance, or 1 every 16 thymine dimers. You'd expect this polymerase to stick in the right pair of nucleotides just by chance. So 15 out of 16 times, you're going to have a mutation, on average. Another thing that causes mutations, and this is a huge issue, not just exhaust and char-broiled hamburgers, but this is the bigger issue of environmental carcinogens. So stuff we breathe in, stuff we're exposed to every day. Those of you that own cars in California have that big sticker on the window that says this car is made of components that are known to be carcinogenic to the state of California. It turns out there's a chemical that's formed, this benzopyrene molecule is formed when you charbroil proteins. That black stuff that some people like to eat, right, has some carcinogen in it. Maybe a little bit. It's kind of like cigarette smoke, potentially. This chemical, benzopyrene, itself is not reactive. For those of you, which are most of you, some of you are taking organic chemistry at the same time as this class. You haven't gotten there yet. For those of you that have, does this look like a reactive molecule? Is it likely to chemically react with DNA?
Yeah.
Why? This is a pretty stable molecule, actually. It's aromatic. All of its bonds are satisfied. It's not like a peroxide or something like that, which is highly chemically reactive. That's what's exploding down in Texas. The chemical plants that lost their cooling capabilities, and they had a lot of chemicals that are now starting to explode and catch fire. Peroxides, highly reactive. Well, it turns out that one of the ways our bodies deal with this molecule and a lot of other aromatic compounds, these highly planar, multi-ringed compounds, our bodies try to detoxify these when we eat them. That's part of the job of our liver. We have enzymes. Most of them are called CYPs. You do not need to remember this. The names of the enzymes, CYP1A1, 1B1. These are enzymes that start to take that molecule and chemically modify it to make it water soluble so we can excrete it. We want to get these chemicals out of our bodies. It helps if we can make them water soluble so we can pee them out. And one of the first steps in doing that is to make epoxides that can then be reduced into hydroxyls that makes this molecule a little bit more water soluble. Problem is, this first step forms an epoxide. If that epoxide happens to be in your nucleus or get into a nucleus, that oxygen molecule can react and bond with DNA. So again, just like the last story about our error-prone polymerases that are trying to help by finishing replication of DNA, but they insert mutations. Here we've got our liver, part of our body that's trying to help us by getting rid of these toxic compounds, but they accidentally, well, not accidentally, they produce more reactive versions, more dangerous versions of that molecule while they're in the process of trying to get them out of our bodies. So that molecule, the peroxide version, can actually physically, chemically bond with DNA. And when it does, it sticks itself in between, so it reacts with a base pair, it inserts itself between base pairs in the double helix. The only real effect is it kind of makes the double helix a little bulgy. It doesn't quite look like a nice, perfect double helix. But it turns out this has a very similar effect to replication like thymine dimers. The polymerases have a hard time replicating around these benzopyrene adducts when DNA reacts with benzopyrene. And so this can also cause mutations. That does not mean you should not eat charbroiled hamburgers, by the way. It means you should be familiar with the fact that there are some studies that suggest that this is true. Okay, so long story longer, that was the story for last class. What do sunlight and hamburgers have in common? They're both mutagenic. Right, so the French fries I mentioned at the start of class may contain acrylamide, which is also carcinogenic, and mutagenic, but I think the jury's still out on whether French fries are as bad as hamburgers. Personally, give them to me. I'll eat them. Okay, so on to today. I've made a change in the structure of class. We're going to see how it works. I know every class up until this point, we have not covered everything that I've put in the lecture notes, which, again, to me is fine. I'd like you to tell me if you're okay with that, too, because this is about you. It's not about me. But one of the things that's really annoyed me so far is that I've never gotten to the case study. And I'm asking you to read the case studies every class, and you're putting the time in, I hope. But we never actually get to capitalize on that investment in time you make reading the case study. So we do the case study first today. I'm going to do a tiny little bit of looking at these Socrative answers. And then we're going to get right into the case study in the hopes that it spurs you to ask some questions about things that are still unclear. Okay, so I'm hoping, fingers crossed. When a cell undergoes mitosis, how many cells? Okay, good. I'm not going to talk about that anymore. Everybody's known about mitosis, or almost everybody, since high school maybe. So mitosis is one cell dividing to produce two cells. That's how organisms grow. Okay. When a cell, let's see, a human cell, how many copies of DNA of every chromosome are in a human cell? 2. We're diploid. We have 2 copies of every chromosome by definition. So, how many copies of each chromosome should be in the daughter cells? Oops. Two. So over half of you said 2. But yeah, this is mitosis, not meiosis, just to make it clear, in case that's the reason there were a lot of answer A up there. One cell divides. Every daughter cell has exactly the same number of chromosomes as the parent cell. That's the whole point of mitosis. So here's mitosis. Where does synthesis happen? Where does DNA replication happen on this chart of the cell cycle? What does S stand for? Synthesis. So that's where DNA replication happens. So before replication happens, that's G1. If we were human, what's N? N is the number of different chromosomes we have.
23.
We have 23 pairs. So we have, if this was a human cell, at that point in the cell cycle, we would say there are 23 different types of chromosomes, chromosome 1, chromosome 2, chromosome 3, chromosome 4, up until the 23rd chromosome, XY, or XX if you're female. So 2N, twice that, is 46. That's how many pieces of DNA are inside the nucleus during interphase. After synthesis, what happens during synthesis? If you explain it just in plain English, no terminology from genetics. What happens from before synthesis to after? The number of chromosomes doubles. So we should now have an N of N is still the same. How many different types of chromosomes are in the cell? There's still 23 different chromosomes. There's still chromosome 1, chromosome 2, chromosome 3. So N does not change.
It is duplicated.
What changed was the total number of chromosomes, which has gone from 46 to, yeah. So the post-synthesis cell has 92 chromosomes, right? We started with 46 here in G1. After doubling, that's 92. So before synthesis, 46, after synthesis, 92. But N is still the same. We still have 23 different types of chromosomes. Chromosome 1, chromosome 2, chromosome 3, et cetera, chromosome 22, chromosome 23.
So there's still only 46 centromeres, though, for what you're calling in 92 chromosomes, right?
So-
Just duplicated
What is a chromosome?
chromatids, different
Well-
chromosomes, right?
Yeah, chromosomes, chromatids, same thing. So what's the ploidy? This is a vocabulary term from last time. If you have 2N, twice, 2 sets, 2N over here, what do we call that? Diploid, because di means 2. We got 2 sets of our chromosomes. Normally our cells are diploid. 2n equals 46. If n is 23 over here in interphase, what's the ploidy? How many sets of 23 chromosomes do you have if you've got 96?
You should still only have 2.
96 divided by 23.
They're duplicating. Tetraploidy seems like a really rare genetic issue.
Tetraploidy is not at all a rare genetic issue, I'm here to tell you today. You'd think it was, but it's not really. And this is where a lot of the confusion comes in about looking at chromosomes and figuring out what you're drawing when you draw a chromosome and how many copies of chromosome there are. So if we don't get to anything else today, this is by far one of the most important things that you can learn this semester, which is why I take this particularly slowly if there are questions. Before synthesis, there's 46 chromosomes. A chromosome is just a double helix. So if you replicate all of those double helices, you get to 92. You get 92 chromosomes until when? We've got 92. Then you enter mitosis with prophase, metaphase. We're still dealing with a single cell. It still has 92 chromosomes. Here, what happens in telophase? Those chromosomes start to segregate into the 2 daughter cells. So now we're dividing what? On the left side, how many chromosomes are on that part of the cell that's going to become a daughter cell? 46. And here's the other 46. And then after cytokinesis, we've gotten back to, so there's 46 chromosomes in that nucleus, 46 there. We end the cycle back where we started. 46 chromosomes. Right. I mean, it's kind of straightforward. You start with a certain amount of DNA, synthesis doubles it, cell division halves the doubled amount, so you get back to the same number of chromosomes you started with. That's the whole point of mitosis. You start with one cell, you wind up with 2 cells that have exactly the same amount of DNA that you started with. All the cells are identical, almost. But there's this really weird thing about that tetraploid part after synthesis. Our cells, after DNA synthesis, have 4 copies of every chromosome. You have to do that if you're going to divide those 4 into 2 cells that have 2 sets.
I disagree with you.
At some point, we have to have 4.
I disagree with you.
That's fine, but it's a fact.
The chromatids are double, not the chromosomes.
But
You
they're
still have-
all,
you still
so
have 46 chromosomes.
there is
Right,
no
first copy.
difference. I appreciate this.
Yeah,
This
so
is,
I want to challenge this because I don't agree with
so there's nothing to disagree with in that this is fact.
I
The
got
problem that we're
2
having
websites
is,
right here that are disagreeing.
the problem that we're having is terminology.
Yeah, chromatid and chromatin.
Chromatid and chromosome.
Chromosome, they're different.
They're different words, but they both refer to the exact same physical object.
That's not true.
There's no difference between a chromatid and a chromosome. Hang on. What's this?
Single strand of DNA.
That's 2 strands of DNA.
Double helix.
That's what do we call this? It's double helix. It's also is it a chromosome or a chromatid? It's a chromosome. So, this is great, by the way. This is a perfect question for this point in time. Now we're going to have, so let's say this is chromosome 1, human chromosome 1. And it's the version that you got from your dad. So I'm going to put a P there. This is a nomenclature I use that nobody else uses. I invented this. 1 paternal, that's the chromosome 1 that came from the dad at fertilization. So there's also going to be a copy of chromosome 1 that came from your mother at fertilization. So we're going to call that chromosome 1M, 1 maternal. Diploid. Okay. At what stage of the cell cycle? We were just looking at a diagram of the cell cycle. You can flip back if you want to and look at it. At what point during the cell cycle are our cells diploid? They have 2 copies of every chromosome.
At every stage of mitosis. At every single stage of mitosis, the cells are diploid.
No.
Yes, they're only haploid in meiosis.
So this is a perfect opportunity, just like the exercise I asked you to complete for today. This is another version of it. These are all chromosomes, or chromatids, double helices, from the same organism. This is my favorite organism, the muntjac. Google it. There's lots of cute pictures of muntjacs. You spell it like this, if you're not familiar with what a muntjac is. It happens to be the mammal that has the fewest number of chromosomes, so it's an easy case to talk about in this context. Okay. At what point during the cell cycle do we define ploidy for an organism? G1, before synthesis. So here's an interphase G1 nucleus. I'm going to circle it in red. And this is really cool. What these geneticists did was they use a fluorescent dye that specifically binds to the different chromosomes that the muntjac has. So you can count, basically what I'm telling you is you can count colored blobs, and that tells you how many different chromosomes are in the nucleus. So you can see down below at metaphase, that's during mitosis, you can see the chromosomes, chromatids. Right? After synthesis, they're chromatids. So how many copies of chromosome green, that is green, right? There's green, blue, and red chromosomes in the interphase? Is it close enough? Okay, help out the colorblind guy. So how many green blobs are there in the interphase nucleus? So think about that while I take the question, not an answer.
Okay. I was just wondering, for each of the chromatid or chromosome X's, is there 1 DNA or 2?
That's 2. Quick answer, but that's exactly where we're heading, is how to look at those metaphase chromosome spreads and decide how many double helices are in that picture.
So how many chromosomes would you say are in that picture, that
Which-
karyogram?
The one on the left.
Yeah.
This is how many, sorry, let me make sure I got the
How many chromosomes are in that chromosomes
chromosomes.
in that karyogram?
So
Your
one
answer.
of the questions, my answer is long-winded.
There's only one number.
So I'm Socrative. I don't just use Socrative. I also use the Socratic method, which is I answer questions with questions. What stage of the cell cycle are we looking at here on the left?
Metaphase.
Anytime, exactly right. Anytime you see chromosomes as discrete objects, that's the point of the slide on the right. Anytime you can see something that looks like something we call a chromosome, not just a blob like those interphase blobs, when you see something as a line or an X or a Y or something like that, I guarantee you're looking at metaphase. That's the only time during the cell cycle when the chromosomes are compacted. So at metaphase, that's after synthesis.
That's right.
So now we're
under your
talking
definition,
about,
it should be tetraploid, right?
so now we're talking about chromatids.
I'm talking about chromosomes.
The definition is, after synthesis, there are only chromatids until cell division completes. So these are chromatids, but remember, a chromatid
So those aren't chromosomes anymore?
is a double helix, and a chromosome is a double helix. The only difference between them is what time during the cell cycle you're looking at them. So this is metaphase, which is post-synthesis, so we're looking at chromatids. Now I'm going to number them for you, and we're going to answer your question. That object that I'm circling right there, that's a double helix.
It's a single chromatid.
That's one chromatid. There is a second chromatid.
That's right, sister chromatids.
So that's one, that's two, sister chromatids.
Yep.
and they're connected
Centromere.
at their centromere.
Right. My point is, that is a chromosome.
No,
That is one chromosome.
that is,
Oh, man.
for this class, the point that is most confusing, and you certainly understand it, all of you by now, is that the X-shaped thing is 2 double helices.
They're identical copies.
They're identical copies or almost identical mutation. So I've circled, so yeah, for all intents and purposes, we'll say those are identical double helices. If this was the muntjacs parents, we would call this, say, chromosome 1 from the dad, and the copy, the new version of chromosome 1 from the dad. Who can find chromosome 1 from mom? Where is it in here? It's the biggest one that's on the bottom. So we've got chromosome 1 from mom, and we've got a chromosome chromatid 1 from mom. 2 of them, joined at their centromere. If we blew this up even more, which I can do thanks to the power of whatever program this is, if we looked in detail at this, each of those objects is a double helix. And there are 2 of them lying side by side. So that's a pair right here. That's a pair of sister chromatids.
Yep.
It's the version from mom. And there's a pair of chromatids. But we do not, and this is definition.
And you're calling these tetraploidy.
This is currently 4
4N?
copies, 4N. This is currently 4 copies. There are 4 double helices up here, right, of chromosome 1.
Man, because in the first slide, we had that exact same picture, and you told us it was 2N equals 6. Remember the
It
first
is
lecture we had?
because, okay, I move on, but let's answer that question, not right now. Think about this. This will be your reflection question for next class, is how can we reconcile these 2 views? Earlier, I did, you're absolutely right. I put up this slide, and I said that this is a cell from an organism that is diploid, and 2N equals 6.
That's right.
I did. I said that.
6 chromosomes. Not 12.
So who knows the answer?
I would guess 12.
Exactly right. We defined ploidy before synthesis. That's the 2N equals 6.
Okay,
Here,
synthesis, right?
this is after synthesis, so we have how many double helices in that red circle? There are 6 objects. Each object is made up of 2 double helices. So this is
12 chromatids,
12 chromatids.
6 chromosomes.
This, last point I'll make about this, we'll have to have offline conversations otherwise. This thing, the X-shaped thing that you see in a picture, that's not, we colloquially call that a chromosome. We're used to saying that thing is a chromosome. We look at it, we see what looks like a single object. We refer to it as an X, single. It's not. It's 2 overlapping or touching or next to each other double helices that look like a single object, and so we're used to calling it a chromosome, but it's 2 double helices. Outside of metaphase, or outside of mitosis, we would call those chromosomes. Inside mitosis, we call them chromatids. Regardless, it's 2 double helices. Now, fortunately, we can move on and talk about this, which has nothing to do with the content we just talked about in terms of having to define ploidy. And hopefully you'll find this particularly interesting. I think it's got a real interesting real-world relevance, this story. Single human oocytes. Did anybody figure out what the authors, there's a real-world problem the authors are trying to solve here. This is a long paper. I certainly appreciate it if you didn't pick it up. If you read all the way to the discussion, which I didn't ask you to read, maybe you found it. So here's the question. You know we can do in vitro fertilization, right? How does that work? You take the egg out of the mother, you bathe it in sperm, or you put one sperm on it. Depends on how you're doing it, and you get an offspring. Okay. One of the reasons that in vitro fertilization could be really useful, especially in the genomics era, is because we could try to genetically test. This is the goal. You want to be able to genetically test oocytes to figure out which ones maybe have some mutations that you want to avoid. So it's not like you'd want to use any oocyte for in vitro fertilization. You might want to find the one that you know has, say, the right number of chromosomes. So how many chromosomes, we're talking about humans, of course, not other species. How many different chromosomes do we have in our karyotype? 1, 2, 3, 4, 5, up to 23. So each of our germ cells, both a sperm and an egg, should carry one complete set of chromosomes, so that when they fuse together and make the offspring, you've got 46. The problem is that sometimes, a lot of times, this doesn't happen. This is one of the major cases of spontaneous causes, of spontaneous abortions, when an embryo doesn't have exactly 2 of every chromosome. That will often cause spontaneous abortion. And it was only recently that we learned this. For probably obvious reasons, most people that have spontaneous abortions don't want to share that news. They don't want the fetus to be studied. It's emotional. And so there wasn't a lot of research on this until recently. So the question is, genome analyses of single human oocytes. How do we tell whether or not an oocyte that's about to, an egg that's about to be used for in vitro fertilization has one copy of each of the 23 chromosomes? How do we know if it's a good egg or not? Good egg, bad egg. Okay, perfect. So what tools do we know about so far that we could use to answer that question? You could make a karyogram, right? We could look and see, does that oocyte have a copy of chromosome 1, one of chromosome 2, and so forth. Except what?
Well, once you see the DNA, you can't quite get it back into the cell.
Very problematic. Once we take the DNA out of the oocyte to look at it, we destroy the oocyte. So, oh, it was a good oocyte. Shit. Let's try another. Oh. So the question, the real world application here was, how do you genetically test an oocyte for quality, that is at least the presence of all the chromosomes, without destroying it? Now, lest I make another mistake, or at least more confusion, I want to point out right now that I have a big bias in how I view meiosis. And it's not good. I'm going to see if you share it with me. I asked, when you have a cell undergo meiosis, how many cells are produced? And you should have answered 4. But that's if you share my bias. Because we've all heard that meiosis is the process of one cell dividing and dividing again. So you go from 1 to 2 and you go from 2 to 4, that's meiosis. That's how it counts out. But what I asked you to focus on for this manuscript was there is an important difference in what those cells are between males and females. So if you're a male, 4 is absolutely right. It's not wrong if you're female, but one cell divides into 2, 2 cells divide into 4, sperm. All 4 of those sperm are capable of activating and fertilizing an oocyte. What's different in females? Polar bodies. So the first cell division in this is not true in males, this is only for females in humans. The first cell division is asymmetric. So that first cell division, you get what's called a secondary oocyte, and you get a polar body. So that first cell division, you start with a cell that has, as shown up there, 4 blocks of DNA. We call those chromatids. That first cell division divides them in half. So we've got 2 there, and we've got 2 there. What happens to that first polar body? That's the first division of meiosis, an asymmetrical division. The big one is going to be the one that produces the oocyte. The smaller cell that's a product of that first division is called a polar body. It doesn't do anything. Okay. So what happens to that primary oocyte, or the secondary oocyte, rather? The one that's going to divide, and one of the daughter cells will be the oocyte. It divides, it's asymmetric again. What happens to the cell that doesn't become the oocyte? That's what we call the second polar body. And then you've got the oocyte. This is not how it works in guys. Guys, it's straightforward, the way we expect. 1 division, second division, 4 cells. So how many cells does a female produce after meiosis in humans? The oocyte, the first polar body, and the second polar body. And that's what's diagrammed here down at the bottom. We've got polar body 1, we've got polar body 2, and we've got the oocyte, which is called female pronucleus here. This is how the DNA is divided up among those cells. There are 2 copies of every chromosome in the first polar body. There's 1 copy of every chromosome in the second, and there's 1 in the oocyte. The oocyte is the thing we want to fertilize if we're going to do in vitro fertilization. So we start meiosis having 4 copies of every chromosome. We wind up with an oocyte that has just 1. So hopefully everybody's good at addition. 1 plus 1 plus 2, simple math, 4. How does this help us solve the problem? Can you do subtraction? Ask a stupid question, get a blank stare from the audience. If you can do counting up how many chromosomes should be in those 3 different cells, what if I told you that the goal was to figure out how many copies of a chromosome were in the female pronucleus? And I told you that there were 2 chromosome 1s in the first polar body and 1 chromosome 1 in the second polar body. Then how many would be in the nucleus of the actual oocyte? You start with 4, 2 are in the first polar body, 1's in the second polar body. 4 minus 1 minus 2 leaves you the answer, which should be, ideally, in most circumstances, 1. So what do the scientists do? They know that every oocyte before meiosis starts with 4 copies of every chromosome. So instead of destroying the oocyte, what do? What was that? They take out the 2 polar bodies and they count how many chromosomes are in the polar bodies. Simple math. You start with 4, How many copies of the chromosome are in the two polar bodies total? There should be, normally, 3 total in the two polar bodies. 2 plus 1. That's if meiosis is working correctly. If it's not, what would happen if I said, oh, we found a case where in polar body 1, there were 2, and in polar body 2, we found another 2 copies of the same chromosome. How many are in the oocyte? 0. Don't use that oocyte for in vitro fertilization. So that's all this paper was about, was a proof of principle that what you could actually physically take develop a cell undergoing meiosis, remove the 2 polar bodies, take the DNA out, count it accurately, and predict the genotype of the oocyte. That's right. If it's complicated, very complicated. So the last thing we need to know about this paper then is how to interpret this graph. But first, sir.
So how would they realize whether the 2 polar bodies in the oocyte were all originated from the same cell, or primary oocyte?
So they have to take these out of a person at the time when meiosis is starting. So you can actually, like that cartoon on the previous slide, if you take out the oocyte, the polar bodies are still attached to it. So you can know that those all originated from the same process of meiosis, that they all started from the same single cell before it started dividing to produce the gamete. So a few pretty straightforward questions. Bless you very much. Can you say that? Bless you very much. What's on the x-axis here? This is a plot of DNA content of the first polar body, the second polar body, and then the pronucleus, the actual oocyte. So in this case, what they did was they actually did destroy the oocyte. They weren't trying to use these for fertilization. They wanted to test the hypothesis that you could do simple math with gametes. So they took the first polar body, the second polar body, and the oocyte, and they took the DNA out of all of them. They wanted to make sure that 1 plus 2 plus 1 equals 4. So what's across the bottom here?
Numbers.
Yep. They're representing the chromosomes. So we've got chromosome 1, chromosome 2, chromosome 22, and because this is a female, they're looking at the X chromosome. Up the y-axis is DNA copy, or the number of copies of chromosome. So that's basically DNA content. How many copies of each chromosome there are in each of these cells. If you tip your head and look like that. Okay. So, first polar body is supposed to have, if the cell biology follows that cartoon, the first polar body should have 2 copies of every chromosome. So what does this show? Is that true for all of the chromosomes?
Not 16.
So not 16. We've got 3 copies of 16, 3 copies of chromosome 22.
19. In the beginning part of 2.
And then we've got too little of chromosome 19. We've got 1 copy of chromosome 19. Part of, over here on the left, not all of chromosome 2, but part of chromosome 2, there's too much of. Right? This is the first time scientists have looked inside a gamete and the polar bodies to look at how much DNA was present in a gamete, a sperm or an egg, in this case an egg, oocyte. Okay, so that's not good, right? Should be 2 copies of every chromosome in the polar body, yet it's missing some, and it's got extra copies of some parts of some chromosomes. Okay, second polar body. Is it looking like we expect 1 copy of every chromosome?
Almost.
Almost, except for there's an absence of any part of that part of chromosome 2. Shocked?
That's where it came from.
Exactly. So
As much at the
it's,
beginning as you do at the end.
yep. The one polar body has a little bit extra part, so that chromosome broke. Part of chromosome 2 broke and stayed in the first polar body. That's why the second polar body is missing it. So one cell has too much, another cell has to have too little, because this is math. You start with 4 of every part of every chromosome. It has to total 4 at the end.
Why isn't that the same for the chromosome like 16 or 22?
So chromosome 16, we just have to make sure that in the 3 cells we're looking at, there's 4 of all of them. So when we look at the actual, circling here in black, female pronucleus confirmed, that's when they destroyed the oocyte and looked at its DNA content. Would you use that oocyte, last question, would you use that oocyte to fertilize? Female pronucleus confirmed. We could. Is it ideal? Does it have one copy of every chromosome? No. It's missing, it has 0 copies of chromosome 16, it has 0 copies of chromosome 22, and it's got an extra copy of chromosome 19. But they would have guessed that. They would have calculated that, even if they hadn't destroyed the oocyte to confirm that that was the genotype, because there were already 3 copies of chromosome, what was that, 16 in the first polar body, and there was a 4th copy, so that's 3 plus 1. They'd already found all 4 copies of chromosome 19 in the polar bodies. So there had to be none in the oocyte. They just confirmed that by looking in the oocyte, and it's true. And we saw exactly the same thing for chromosome 22. There were too many total copies in the first 2 cells, the ones that don't make gametes. So it had to be the case that the oocyte didn't have a copy of that chromosome. There's 3 copies of chromosome 22 in the first polar body. There's 1 in the second polar body. It leaves none for the oocyte. So the more we look into this as scientists, the more often we find that, yeah, chromosome segregation and making sure that the right number of chromosomes winds up in these cells that are going to make humans eventually, this is not always really robust. That is, it doesn't always happen right. More often than not, there are these sorts of chromosomal differences that you wouldn't expect. So any questions about this? So all it boils down to is simple math. If you start with 4 chromosomes and you look in the 2 polar bodies, you can figure out how many are going to be left in the oocyte. 4 minus something gives you the answer. And then you don't have to destroy the oocyte. All right. So I had 2 different papers I was thinking about asking you to read for next class. Just look at one of them. Just read the one whose first author is Butler. It's really short. Next class, we're going to be talking about alleles. That is how we use terminology to distinguish the paternal version from the maternal version of a chromosome, of a gene. There's some nomenclature involved, but we're going to make this very relevant to society because what this paper is about is basically genetic profiling. It's how can you tell whether or not, by their DNA, whether somebody's from a particular human population. African American, Hispanic, Caucasian, what have you. So we're going to be talking about how scientists do this, the technique of polymerase chain reaction, and the application of that technology to essentially genetic profiling, fingerprinting. And I'm going to send you on Google Classroom something to try related to the technique of PCR. And with that, I'll see you next class. Please, please, please come to office hours to ask further detailed questions.